All right, let's jump right into this explainer. If you've ever found yourself wondering about the exact microscopic science that actually goes into assisted reproduction, you are absolutely in the right place. Today, we're going to give you a clear, chronological mental map of what actually goes down inside the IVF Embryology Lab. We're tracking the complete journey, right from the moment gametes are collected all the way to the point an embryo is transferred or safely frozen in a tank.
For so many patients, you spend a relatively short amount of time in the actual procedure room, right? And that leaves the lab itself feeling kind of like this mysterious black box. You hand over your hope, you go home, and days later, you get a phone call. But today, we're going to completely demystify that hidden sequence. We're going step by step, minute by minute, dish by dish, and incubator by incubator.
Now, I absolutely love this quote because the lab's job is not to magically, force life to happen. That's not how it works. Instead, it's meticulously optimized to mimic the human body. The embryologists are basically trying to create the most perfect, highly regulated conditions possible so that biological development can just take its natural course outside of the body.
So, here is our roadmap for today. We're going to cover inside the IVF laboratory, the arrival of eggs and sperm, the moment of fertilization, growing inside the incubator, embryo grading and selection, and finally transfer, freezing, and beyond.
Okay, let's start with Section 1, inside the IVF laboratory. Before a single egg or sperm even enters the room, we have to understand that this lab is a highly controlled biological environment. Think of it like part operating theater, part high-tech cell culture facility. We're talking about incredibly strict temperature regulations, exact pH balances, and air that is literally scrubbed clean of volatile organic compounds, or VOCs. Why? Because small deviations in literally any of these can significantly affect outcomes. The embryos are incredibly sensitive to their surroundings, so the environment has to rigorously mimic your natural physiology.
Moving right along to Section 2, eggs and sperm arrive, the day of retrieval.
So when the clinical team hands the samples over through the lab window, they aren't passing over like neatly isolated, perfectly clean eggs. Instead, the lab receives tubes of follicular fluid. Embryologists have to immediately take this fluid, keep it perfectly warm, and embark on a microscopic scavenger hunt to find what we call cumulus oocyte complexes, which in plain English is an egg hiding inside a tiny protective cloud of cells.
And this brings us to a really massive point of confusion. Seeing a certain number of follicles on an ultrasound does not automatically equal that exact number of mature eggs. The very first question the lab asks isn't, "Is this a good egg?" but simply, "Did we even recover an oocyte from this fluid?" Remember, we are collecting opportunity, not certainty. Some follicles end up being empty, and some of the eggs we do find might actually be immature and completely unable to be fertilized. Meanwhile, on that exact same day, the other half of the biological equation is being prepped. Raw semen is never used as is. It has to be washed and processed. The lab uses techniques like density gradient centrifugation to strip away the seminal plasma, debris, and, the sluggish sperm. We are intentionally isolating the most highly motile sperm population. We're essentially picking out the strongest microscopic marathon runners for the next critical step.
Which brings us to Section 3, the moment of fertilization. Once both the gametes are prepped and ready, we hit a major fork in the road. How will fertilization actually happen? Well, there are two main pathways. Conventional IVF is exactly what it sounds like. Prepared sperm are added to a dish with the eggs, and fertilization is just allowed to happen naturally without direct microscopic manipulation. But on the flip side, we have ICSI, or intracytoplasmic sperm injection, which takes a much more hands-on approach. In the ICSI procedure, the embryologist first strips away those protective cumulus cells to physically check if the egg is fully mature. Then, using these crazy microscopic glass needles controlled by incredibly precise joysticks, they carefully immobilize a single, visually selected sperm and inject it directly into the mature egg. It's an absolute stunning feat of micromanipulation, usually used for severe male factor infertility. But even with this tech, you've got to remember that simply placing the sperm inside the egg doesn't guarantee fertilization. Biology still has to take over.
All right, Section 4, growing inside the incubator. This is the agonizing waiting period for patients.
Picture this: tiny little droplets of specialized nutrient media, covered in oil to prevent evaporation. The embryos are growing inside these tightly sealed, dark, super warm incubators. Now, the embryology team actively minimizes how often they open these incubator doors because they want to protect that delicate environment. But they do check on the embryos at specific intervals, typically day 1, day 3, and then between days 5 and 7, just to track their major developmental milestones. The very first of those milestones hits on day 1. The embryologist is specifically looking for the appearance of 2 pronuclei, often just called 2PN. Seeing those 2 little circles inside the egg, that is the ultimate visual confirmation of normal fertilization. It physically proves that we've got 1 maternal and 1 paternal genetic contribution present. It is an incredibly promising sign, a fantastic phone call to get. But it's still just the first hurdle in the embryo's marathon. Okay, Section 5. Embryo grading and selection. As we get to days 5, 6, or 7, the embryo hopefully hits the blastocyst stage. The embryologists now have to assess them to figure out which ones to prioritize. They use precise visual criteria for this. For early embryos, they're looking at cell number, if they're symmetrical, and if there's fragmentation. For blastocysts, they look at the degree of expansion, the tight bundle of cells that will become the fetus, called the inner cell mass, and the outer layer of cells that becomes the placenta, known as the trophectodote. Now listen up, because there is a massive myth surrounding this stage. So many people think a perfect grade equals a guaranteed baby and a poor grade equals failure. Actually, scratch that. Morphology isn't perfectly predictive at all. A beautiful, highly graded embryo might fail to implant due to unseen genetic reasons, while a totally average-looking embryo could result in a healthy, successful pregnancy. Grading is really just the best selection tool the lab has to prioritize which embryo to transfer first. It is definitely not a crystal ball for destiny. Section 6. Transfer, freezing, and beyond. The home stretch. Depending on the patient's specific treatment plan, the journey branches here. One highly graded embryo might be carefully loaded into a microscopic catheter for an immediate fresh transfer back into the uterus. Or the embryo might undergo a safe, ultra-rapid freezing process called vitrification. Alternatively, it might be biopsied, where a few cells are safely plucked from that outer trophectoderm layer for pre-implantation genetic testing. After that, the embryo is frozen while everyone waits for the genetic results to come back.
And speaking of freezing, far from being just some backup plan for leftover embryos, freeze-all strategies are incredibly common and intentionally utilized today. Modern cryopreservation is just astonishingly effective. It's used for fertility preservation, to allow time for genetic testing, or simply to ensure the patient's uterine lining is in the absolute most optimal natural state to receive the embryo in a later cycle. It's a game changer. Throughout this entire sequence we've covered today, there is a totally unseen backbone holding the whole shebang together. Excellence in an IVF lab isn't just about having steady hands for micromanipulation. It's about relentless systems. Strict identity witnessing, using both human double checks and electronic tags, alongside rigorous daily quality control of gas lines, temperatures, and incubators. All of this ensures that the wrong thing absolutely never happens, and the perfect environment is always maintained. So, this explainer has given you the foundational mental map of the entire embryology journey, from follicular fluid arriving at the window all the way to an embryo safely frozen in a tank. Knowing just how incredibly delicate this balance of biology, timing, and precision really is, it begs a pretty fascinating question. What hidden marvels are we going to uncover when we zoom in closely on the exact moments of ovum pickup next time? Join me for our next explainer, where we will peer directly into the microscope to see exactly what the embryologists are searching for.